All right, my lovelies. Good morning. I am trying out this whole filming on the document camera since I'm not here with you guys today for the replication fork. So you're going to have to bear with me. This is the first time we're trying this. But I wanted you guys to be able to get your DNA replication big diagram done while I was gone, and I thought that it would be helpful if I recorded a video of me going over the replication fork diagram so that we could kind of all be on the same page when I get back, and we can pick this up on Monday. So I've left with your substitute, I've left your blank copies of your diagram that you need to put together. I've left my copy of the answer key, and I've left the written copy of, like all the stuff that I write in all these little boxes. I've left that written copy too, so that if you're like, oh, Miss Latner, I can't see that close up, when I start to zoom in, you can totally go grab that from your sub and use it. But I wanted to go through this with you real quick. So I'm going to start over here at the beginning of the diagram. This whole diagram is what we call the DNA replication fork, and this is the site where DNA replication is going to start in the replication process. And so let me orient you to what you're looking at right here real quick. So we start out with the DNA double helix, and you can see that the double helix is made of base pairs, and those base pairs are of course A to T and G to C as always. And this double helix is wound into its helical shape. We have some labels here at the end. Let me zoom in for you so you can see a little bit better. For the DNA strand, we have our 3' and 5' end labels. Remember that one strand is going to begin in 3 and then end in 5 down here. And then the other strand is going to begin in 5 here, and then of course end in 3 here, because our DNA strands are anti-parallel, meaning that they run in opposite directions. And so we begin replication at what is called the replication origin, and this is an AT-rich sequence, so an adenine and thymine-rich sequence in the DNA that is easy for an enzyme called helicase, that's this orange one right here, to break apart. And so helicase is the enzyme that kind of starts off replication, and it does so by breaking the hydrogen bonds that hold the two strands of DNA together and creating what we call the replication fork, which is this open area here. This open area, as helicase is moving in this direction, opening up more and more of this fork, it creates all of these twists in the DNA strand and the part that's still in double helix. And so we have a second enzyme right here called topoisomerase, and topoisomerase is responsible for undoing some of this twisting and making sure that the DNA strand doesn't break because it's being hypercoiled or supercoiled. And so really that's our first two enzymes for replication here. We have DNA helicase and then topoisomerase. Helicase binds to the replication origin and begins our replication, or the unwinding of DNA for our replication. It does so by breaking the hydrogen bonds that hold together the two strands. And then we have topoisomerase, which is responsible for cutting DNA and breaking a phosphodiester bond so that some of this hyper twisting or supercoiling that's going on up here can relieve the tension that's caused by helicase moving in that direction. So we're left with this open space here where both of the strands have been separated. And the next thing that comes into play are what are called single-stranding binding proteins. And these are these yellow proteins that you see here. They're responsible for keeping the DNA strands apart. So these two strands would naturally like to hydrogen bond back together. We call that re-annealing. And to keep that from happening, these single-stranded binding proteins, they hop onto each of the strands and keep them from coming back together or closing back up. Because those strands have a great affinity for each other or an attraction for each other because of hydrogen bonding. So the single-stranded binding proteins, they are responsible for keeping the replication fork open so that we can actually do DNA replication. We have this big open area that we call the replication bubble. This is where all the action happens. This is where replication actually occurs. And floating around this replication bubble, we have DNA nucleotides, so that would be A, G, T, and C, and they will be free floating so that when we get to the point where we're ready to add them on for replication, they're just kind of there hanging out all together anyway. And so that's all kind of going on in the replication bubble. You can see that when we separate the double helix and we get these two strands, that these two strands are different. One is called the leading strand, that's this one up here. It's called the leading strand because replication happens in the direction that helicase is moving. So in this example, helicase is moving this way. And so the leading strand, replication's happening in that same direction. It follows helicase. This strand is called the lagging strand. And the reason it's called the lagging strand is because replication occurs in short segments instead of one long continuous line. And it occurs away from the direction of helicase. So again, helicase right here is moving in this direction. So replication on the lagging strand is going in the opposite direction of that. Now, the purpose of, like, why do we have a leading strand on the lagging strand? That's because of this next enzyme that I wanted to introduce, which is DNA polymerase III. DNA polymerase III is the enzyme that's responsible for doing DNA replication. It's the one that does the base pairing so that we replicate our template strand into our new strand, and we do so via complementary base pairing. But DNA polymerase III has a limit. It can only move in the 3' to 5' direction, which means that it can only chase the helicase enzyme on this strand. On the bottom strand here, what is the lagging strand, 3 to 5 is in this direction, so it has to go away from helicase. So we have some slight differences in how DNA replication occurs on the leading strand and the lagging strand. Do not be fooled. The leading strand is not always on top, and the lagging strand is not always on bottom. That's just how it is in this diagram. The leading strand will always be the one that's going towards helicase. The lagging strand will always be the one that's going away from helicase. Other ways that you can tell the difference between leading and lagging is that leading replication is what we call continuous, so it's a single line of replication, and then lagging replication is discontinuous. It happens in these short segments that are called Okazaki fragments. And those Okazaki fragments at the end of replication, they have to be put together via an enzyme called DNA ligase. Okay, so let's start with leading strand replication. So as the replication fork opens up, the enzyme DNA polymerase III is going to bind to the template strand and is going to begin placing nucleotides in the correct place via complementary base pairing. So let's do some for instance here. For instance, if this is my A, then DNA polymerase III puts a T there. If this is my G, then DNA polymerase puts a C there. So it does complementary base pairing. So if this one is a C, cytosine, then G is going to go right here. So this nucleotide is going to come in right there. And so it'll keep doing that until it replicates the entire strand. Now, DNA polymerase III has another limit, and that is that it can't start replicating off of nothing. It has to start replication off of what we call a primer. And a primer is a short sequence of RNA, not DNA, that is complementary to the DNA strand. This allows DNA polymerase III to hop on to this double strand right here and start the actual replication process. The primers are put down by an enzyme called RNA primase. And RNA primase is responsible for making or synthesizing the RNA primers that are complementary to the DNA strands. And that gives DNA polymerase III something to jump onto and then move in that direction to replicate DNA. Now for lagging strand replication, this is going to require a little bit more work. We're going to have several RNA primers. So here's one, there's one. So those are all going to be laid down by RNA primase. We're going to have several DNA polymerase IIIs because this is discontinuous replication. So this DNA polymerase III will hop on right here to this primer and then synthesize the section in between this primer and that primer. Sorry, this section in between that primer and that primer. This RNA polymerase 3 is on and is synthesizing the section in between this primer and this primer. So it goes from primer to primer, synthesizing the new DNA strand. So for instance, again, if this is my G, then it's going to lay down C next. Here's a G, so that'll lay down a C next. And so it'll keep going until it hits this other Okazaki fragment where that primer starts. Now that we have synthesized both of the strands of DNA, we've got a couple things that we still need to do. First of all, we need to make sure that we understand the difference between leading strand, moving in the same direction as the replication fork, and lagging strand, which is going to move in the opposite direction of the replication fork in a discontinuous manner, building DNA, while this is happening in a continuous manner. So once we've replicated these two strands, the leading strand is pretty much good to go. It really doesn't have anything to do to fix it. It just will keep going until we've replicated the entire chromosome. Well, the lagging strand, it's got a little bit of work to do. We have to connect these Okazaki fragments, and we do so via an enzyme called DNA ligase. DNA ligase will form a phosphodiester bond in between each of the Okazaki fragments. So let's take a look at this one. So here's one end of an Okazaki fragment, and here's another, and you can see that they're not connected. DNA ligase is going to go in and make a phosphodiester bond right there and connect these two Okazaki fragments. Another enzyme called DNA polymerase 1, not to be confused with DNA polymerase 3, is responsible for going in and pulling out these RNA primers and replacing them with DNA nucleotides. Because remember that DNA nucleotides, while similar to RNA nucleotides, they are not the same. DNA is deoxyribonucleic acid, whereas RNA is ribonucleic acid. So we've got a slight chemical difference in those two types of nucleotides. So DNA polymerase 1 will go in and pull out the RNA nucleotides and lay down the complementary DNA nucleotides, so it gets rid of the primers for us. And then ligase is going to go in and connect the Okazaki fragments. So we've got DNA polymerase 1 and ligase happening there. The last thing I want to talk to you about is repair. So let's say that we had a mismatch error, meaning that what was supposed to be an AT pair accidentally became an AC pair, so a mismatch. This mismatch is going to have to be repaired. And so we have what's called exonuclease function in our DNA polymerase 2 and DNA polymerase 1 enzymes, and this function will excise any mismatches that we have. And so it goes through and it double checks DNA strand before the replication fork closes up and makes sure that there's no mismatches. And it also will excise and repair any DNA damage like we see with UV radiation. So that's kind of one of the things that we talk about. The reason that UV radiation causes cancer is because it damages DNA. And what do we mean by it damages DNA? Well, it creates these situations where we don't have correct matches on opposite sides of the DNA strand. And so our exonuclease activity in our DNA polymerase 2 and DNA polymerase 1, they're going to be responsible for fixing all of that. Okay, now I know I've thrown a lot at you with this replication diagram. I want you to finish your diagram over to Thursday and Friday this week. We will talk about it on Monday. We'll go over it briefly to make sure that we're all on the same page about it. Then, as usual with these BIG diagrams that we like to do, this needs to be your bread and butter until this test. You need to study it, study it, study it. Hang it somewhere where you'll look at it all the time and make sure that you understand the process of DNA replication. Make sure that you can use your diagram to explain the process of replication, and especially explain the difference between leading strand and lagging strand replication, because they're different. All right, I'll see you guys on Monday. I can work out any misconceptions that you have, and we can fix anything that we might have made a mistake on, but you need to work on this diagram for Thursday and Friday. Thank you.